Bom dia a todos. Vamos começar. Desculpa-me do atraso, que tive um pouco mais ambicioso depois de começar, por causa da transmissão online que a gente estava providenciando. De qualquer forma, eu apenas para agradecer, né, a decana por se gentilmente abraçar-nos desde o início, né, para fazer esse evento ocorrer. E depois agradecer a Terezinha também, que ajudou bastante, e o superintendente geral do CARI, o André Macrae também, que não mediram esforço nenhum para que esse evento ocorresse, né? Então, sem demais longa, vou passar a palavra à decana. Obrigado.
Okay, I will talk in English because to introduce Dr. Fischer. Good morning, everybody. Thank you for your patience. And I will call Fernanda, that will do the presentation of Professor Alan Fischer for us, and then he will talk for us. I thank you for coming to UFRJ. It's very nice to receive you in our Center of Science of Health, and I will pass the word to Fernanda. Thank you.
Bon, bonjour à tous et à tous. Thank you. Professor Alan Fischer, in fact, he doesn't need a presentation because everybody in the world knows him. I need to tell that Professor Alan Fischer is a reference for everybody, not only for immunologists, clinical and researcher immunologists, but I think he's a reference for every doctor in the world. Professor Alan Fischer works for a long time as a doctor and also as a professor and researcher in the Hospital Necker d'Enfants Malades in Paris. And now he's the president of Academy, as everybody knows. And I need to say some words because I have the great opportunity to follow him for about 2 years. And the most incredible thing is that he's a very famous and important professor and important researcher, but he's also a very kind and simple person, and he works with a very enthusiastic way, very kind with the patients, and he always look to improve the knowledge of everybody. So, as I said, he's a very important doctor, and he take care of the patients of all the Europe and also of patients in the Africa and all the world. So, thank you, merci, to be here with us. It's very good to have you here. Thank you.
Thank you, Fernanda. Thank you, Fernanda, for your very kind words, and thanks for your hospitality in this place. I'm pleased to be with you this morning in this nice room. So what I will try to do is to review with you briefly, of course, a field that has emerged over the last 25 years in medicine, that is gene therapy. I think everybody knows what is gene therapy. So the concept is to introduce genetic material, genetic information into cells in order to have a therapeutic effect. So the concept was born with the definition and understanding of the first inherited diseases, but then, as you will see briefly, it became applied to other fields of medicine like cancer, although I'm not going to touch much about cancer. So if you can move, thank you. So the history starts more or less in 1972. That paper, for the first time in Science by US colleagues, they suggested that gene therapy could be considered to treat genetic diseases. So by introducing a normal copy of a gene into the cells of a patient with a given genetic disorder. So that was the time, the 70s, where molecular biology became available as a tool. It was the first time at which cell transfection in vitro, at least, of course, were performed, so, and the first genetic diseases were deciphered. So, but still, this was really the infancy, because since 1972, it took almost 30 years, actually exactly 27, before the first clinical results were obtained. So the next slide. This slide is a summary slide. So if you can, after that slide, if you wish, you can go to sleep, because that's the summary. So there are two ways of performing gene therapy. The classic one is what is called gene addition. So by using a vector, which is a modified virus, either an AAV, adeno-associated virus, or a retro-lentiviral vector, you introduce the cells, which is symbolized by the black rectangle, into cells. There are major differences between the usage of these two vectors. With the AAVs, the genetic material, again, the black rectangle, will persist in cells as an episome in the nucleus, so it will not be replicated when the cells divide, which means that AAVs-based gene therapy can only be used to treat, to modify post-mitotic cells, like neurons or hepatocytes to some extent. Whereas with the lentiviral vector, so based mostly on HIV, modified, of course, and made not toxic, for sure. So in that case, the genetic material is, as shown, integrated somewhere in the genome, although this is not something that we can completely control. So that is, the insertion occurs not randomly, but semi-randomly, which in the first steps of gene therapy was a problem. Today, no longer, for reasons maybe I will have not time to discuss. So with either the technique, so with this technique, the second one, it's possible now to consider to transfer genes into cells that are dividing, because since the transgene is now integrated into the genome, it is replicated together with the genome. So this has been applied mostly to target hematopoietic stem cells and to a little bit to skin stem cells, keratinocyte stem cells. So with this technique, you can try to do three things to compensate for a gene mutation, so typical monogenic hereditary disease. You can inactivate a gene or a regulatory element where you want to get rid of a transdominant negative effect or a mutation that is a gain of function, for instance. And third, you can provide a new function with a synthetic gene. This is what is done with in the CAR T cells treatment of leukemias, for instance. Okay, so then, next, please. The second strategy that is more recent is genome editing. So instead of adding a copy of a gene, you will modify the genome in two possible ways. One that works these days, the second one not yet. So the one that works already today is what is called gene inactivation. That is by using the CRISPR-Cas9 system, or another one as a targeted enzyme to cut DNA. But this is, of course, the one that is the much more convenient compared to others. So you can cut the DNA somewhere. So you guide the enzyme, the Cas9 enzyme, with a guided DNA complementary to the sequence that you want to target. And you can create a modification of the genome, an insertion or deletion or whatever, at the time of repair of the lesion caused by the enzyme. And this is a way to inactivate a sequence. And I will show you, if we have time, one example of that. And the last, and more for the future, the last strategy is gene modification, still by using the CRISPR-Cas9 system, but modified, by the way. If I will have time, we can discuss it, the so-called base or prime editing technology. Then you can insert a sequence or modify a base, a single base, if there is a single base nucleotide mutation, for instance. So it's a possible way also to correct genetic diseases or to introduce a sequence of biological and medical interest. Okay, so this is the summary, and now we'll go a little bit into the detail. Next slide, please. So gene therapy is not that easy. Well, the concept is very simple, extremely simple. But the technology is not that easy, because in order to succeed, you have to go through all of the steps that are mentioned on this slide. So this explains, by the way, why between the first attempts in the 80s, it took 20 years prior the first success were observed. So what you need in A, so on the top left of the slide, that the virus, whatever it's an AAV or a lentiviral virus, lentiviral vector, that the material is integrated into the cell and it's not degraded in the lysosomes. So that's the first point. Second point, now once in the nucleus, the material has to persist, because if it not persist, of course, we'll have only a transient effect, which at least for genetic diseases doesn't work. Third, in C, you need to have a level of expression of the gene, so the quantity of messenger RNA that is good enough, that is not too little, not too high. And so it depends on the promoter you are choosing, and that is put into the vector in order to get the appropriate level of expression of the gene. Fourth, there is the problem in D of the potential immune response of the patient against the genetic material, and mostly against the virus, especially the AAVs, because we are all of us more or less have been infected by natural AAVs, so we have some immunity against these viruses that may neutralize the effect, and that may be a problem that is not fully solved. And on top of it, there is a fifth, you have to target the cell population you want to treat. This is sometimes easy, very easy with the bone marrow, much more difficult, for instance, if you want to introduce a gene in some part of the brain, for instance. Okay, so that's about the problems that have to be considered and addressed for a successful gene therapy. Next slide, please. And now here are this is the summary of the successes of gene therapy observed between the year 2000 and now. There may be a few missing, but I'm not sure. So this is based on the literature and reproduction of results. So these are solid data. So in black, these are names of diseases that were treated by ex vivo gene transfer into hematopoietic stem cells or skin cells, for one example. And in blue, this is in vivo gene therapy based on AAV vectors. So I am not going to review all of them, of course. Just to mention, and I will come back to these diseases, it started with primary immunodeficiencies, so in my field, with the so-called SCID, which stands for severe combined immunodeficiency. Then metabolic diseases, anemia, thalassemia, and sickle cell disease, and other diseases were treated by more or less the same technology. And for in vivo gene therapy, the first success came in 2008 with the Leber amaurosis, so with the retroretinal injection of the AVs to correct the gene defect into the pigmentary layer of the retina, for instance. And then you see hemophilia B and A, so that is by targeting the liver. Spinal amyotrophy, this is a neuromuscular disease where the vector crosses the blood-brain barrier. And up to very recently, the Chinese group has been able to correct, at least partially, an hereditary deafness by AAV injection locally into the ear somewhere. Don't ask me where, because I'm not a specialist. And then, in addition to the treatment of these genetic diseases, since 2014, so 10 years ago, the CAR T cells were developed, as you know, I'm sure. And this has been used extensively now to treat a number of B-cell malignancies, acute chronic leukemia, myeloma, and so on. And to finish with the successes, very recently, a German group has shown that the CAR T cells can also be used to control severe forms of autoimmune diseases, with based on the concept that these CAR T cells directed against the B-cell molecule CD19 are killing the B cells, and by killing the B cells, they kill the B cells that make the autoantibodies. And apparently, they got very nice results. So this is where we stand, which is not too bad. Could be better, but this is in 24 years. There are many, many other diseases that people are trying to address by gene therapy, but we are not yet dead. And on top of that, you see that some of the diseases, they are marked with a star. It means that from this gene therapy, a drug has been developed as a product that is available in some part of the world, at least, which is not an easy topic because these drugs are extremely expensive, and this is a real problem. But there are 17 drugs marketed for gene therapy. Okay, if we move forward. So I will mostly talk about ex vivo gene therapy with retroviral vectors, because this is my field that I know better. So just as a summary, these are the characteristics of these retroviral vectors. We started with the so-called gamma retroviruses, and shifted now maybe 15 years ago to lentiviral vectors that are much more potent to transduce cells because they can infect cells in G1, whereas the gamma retroviral vectors can only transduce cells that are cycling, which is a limitation. So both of them have more or less the same characteristics in terms of gene integration, packaging capacity, which is quite large, 8 kbs. And of course, the advantage, I mentioned that already, it can induce the long-term expression of the transgene, provided that the cells are themselves long-term, but which is the case for hematopoietic stem cells, for instance. And the disadvantages, at the end, is the risk by the introduction of a genetic material into the genome to create an insertional mutagenesis up to a cancer. This happened in a small number of cases, but this issue has been solved by modifying the vectors. I will not go into these details unless you wish me to explain. Okay, next slide. So it started with this disease, this SCID I mentioned already. These are rare diseases where T lymphocytes are absent because there is one mutation in a given gene that impair development of T lymphocytes, sometimes of other cells too, like B cells or NK cells. So there are two diseases that have been treated. So this is a schematic representation of hematopoiesis, normal one on the left-hand side. In the middle, this is the so-called SCID X1, so it's an X-linked disease affecting boys, where the T lymphocytes and the NK lymphocytes are absent because there is a receptor of cytokines, so-called common gamma chain, that is not properly expressed. And the second disease that was treated is the one on the right-hand side, which is the deficiency in adenosine deaminase purine metabolism enzyme that results in the full absence of lymphocytes. So these conditions are rare, but very severe. Patients with SCID cannot survive more than a few months because of the risk of infections. There is a treatment by allogenic stem cell transplantation was developed now 55 years ago, which works very well when a donor is compatible, not so well when a donor is not compatible. Then room for improvement came potentially with gene therapy. So the next slide, please. So how it worked, next slide, please. So the way of doing it in a very schematic manner, so the first trial is to harvest the bone marrow of patients, to select the immature cells, the stem cells, because they have the right marker like CD34, and to cultivate these cells with cytokines to make them cycling at that time at least, and then to infect with the virus, the vector. At that time, that is in the year 2000, this was done 3 times in 3 days. Now it can be done in only 1 day. And then the cells are infused back to the patients by IV source. And at the cell level, so of course it's very schematic, but you see the vector. The vector can enter the cells. So RNA of the retrovirus is retrotranscribed into DNA by the reverse transcriptase of the virus. And the material, if the cell is dividing with the gamma retrovirus, can enter the nucleus and be integrated into the genome. And then with the right promoter, you can get expression of the transcript translated into the protein, expression of the protein, So this is the principle of the first trial that was performed starting exactly in 1999. Next slide, please. And the results were pretty good, still good. These are the first 20 patients treated. So since March 1999, up to 2002. In Paris, then in London, 20 patients altogether. And the results today, so with a follow-up that extends to 25 years for the first patient treated, 17 out of these 20 patients are alive with functioning T cells, so 85%, which is reasonably good for that disease. And of course, the important aspect is the stability over the years of the correction, since these patients actually are no longer patients. They are young adults enjoying a normal life. Some of them still require some immunoglobulin because one cannot, by that methodology, fully restore the deficiency in B cell function, but at least they have normal T cell functions. Next slide, please. So this is just an example for the patients treated in Paris. This is the counts of T lymphocytes in their blood. Each curve of the different color represents one patient. And you can see, so on the right-hand side, you have the dotted lines that are the upper and lower limits of normal counts in adults. And you can see that they are more or less in this range with some variation. So the T cells are there. They are functioning. I'm not going to give you the details. But the other cells of the hematopoietic system are not so well corrected, indicating that actually there are very few stem cells, if any, that were corrected, but rather T lymphocyte progenitors. But the T cells are long-lived. So the hope is that the T cells that one can still detect in the blood of these people 25 years post-gene therapy will still be there, hopefully. Of course, we have to wait in another 25 and 50 years. I will not see it, but hopefully someone will. Okay, next up. But this was not completely without problems. So you can go to the next slide. Thank you. So just to give you a historical account to show that it takes time to go with these processes. For that particular disease, the first time it was treated, that was transplantation, that was 1968. The time the gene of the disease was identified, it's between 92 and 93. So it took 25 years before we knew the treatment. The understanding of the physiology of what the gene does, which is important to know before considering a gene therapy, was identified in 96. Now, if you go further, stop. Thank you. That's okay. So the first treatment is around 2000. Now you can go. I'm sorry. In 2 years later, some genotoxic effect was observed in a limited number of patients because the way the gene was driven was able also to transactivate oncogenes. So this led to leukemias in some of the patients. Leukemias could be treated, but still, of course, it was a major limitation of that technology. Thus, in 2010, 8 years later, it took 8 years, to develop slightly modified vectors, the so-called self-inactivating vectors. I will not go into the details. But now it can be used safely. So this was done since 2010. And then in 2020, the protocol was improved by using a little bit of chemotherapy in order to get not only T cells, but also B cells and NK cells from the gene therapy. And today we are with now almost 25 years of follow-up. So you can see how long it takes and the steps, and sometimes going down and up to some results. Next slide, please. So based on these first results, other forms of severe combined immunodeficiencies, so diseases where T cells are lacking. So this is the picture shows a simplified view of lymphocyte development from hematopoietic stem cells. So you have from the top the NK cells, gamma delta T cells, CD4, CD8 T cells, B cells, and monocytes, macrophages, and dendritic cells. There are other such severe combined immunodeficiencies marked by the blue arrow. And I will not mention them in detail, but just to tell you that today there are 3 SCIDs that have been treated by gene therapy and other clinical trials ongoing from another 2. So the next slide just illustrates for ADA. Thank you. So these are data from our Italian colleagues who first treated patients with SCID ADA. So this is a long-term T lymphocyte counts of these patients. And you can see this is not exactly in the normal range in that case, but ADA is a bit more complicated disease. But still, the patients, they have a subnormal number of T cells, and they do well. So this is clearly a success of gene therapy that has been now confirmed in maybe almost 100 patients worldwide. Next slide, please. So from these SCIDs, then the field moved to treat other immunodeficiencies. This is a bit more complicated because now we go with diseases where there is not just an advantage provided by the expression of the transgene. Among them, next slide please, there is the so-called Wiskott-Aldrich syndrome, which is another X-linked disease where there is a progressive decline in T and B lymphocytes, which, by the way, are not normal in functions. And on top of that, and very importantly, they have a problem with platelets. They have a severe thrombocytopenia. So these patients, they suffered from bleedings, as you can see, and immunodeficiency, but also autoimmunity. So the gene is encoding the so-called Wiskott-Aldrich syndrome protein, and that is important to the architecture of the cytoskeleton of blood cells. So gene therapy was developed with the vector, which is now seen, so self-inactivating lentiviral vector, where the cDNA from the WASP gene is under the control of the endogenous promoter, which is shown in the brown arrow here. So this was done with some success, but not perfect. Next slide, please. This is a study we did between Paris and our colleagues in London, where 9 patients were treated. We have a follow-up that extends now to 16 years, which is not too bad. In terms of correction of the immunodeficiency, it works well. So correction of infections, autoimmunity, it's okay. Now, what is not fully okay is platelet counts. On the top, you have the platelet counts of these patients over time. You see that some of them have normalized their platelet counts, but others still have a relatively low platelet counts, which shows that the technology, as it stands, is okay to correct deficiency in the quantity or the quality of lymphocytes, but not strong enough to fully correct a problem of platelet production. So we still have to make a little bit of technological advance to be able to fully correct such diseases. Next slide, please. I'm moving. I'm sorry, I'm going fast because I try to cover several fields. Next, please. So then there has been studies or trials, actually, to treat, but still by the same strategy, that is ex vivo gene transfer into hematopoietic stem cells, so shown by the red arrow on the left-hand side of the slide, to treat metabolic diseases. So here the idea is that if we can get the gene expressed in the macrophage lineage, a monocyte macrophage lineage, we may get the presence of cells with the right enzyme in different parts of the body, including the brain, because of the microglia. So this was done with some success for up to now at least 3 diseases: adrenoleukodystrophy, metachromatic leukodystrophy, and mucopolysaccharidosis disease type 1, the Hurler disease. So I will just, as an example, show you some results for MLD, metachromatic leukodystrophy. Next slide, please. So this is a study performed by our colleagues from Milan. They published the first study, the first results now a bit more than 10 years ago. Here they did something smart. So but they took a risk, but the risk, that was okay. So they increased quite significantly the quantity of vectors used to transduce the hematopoietic stem cells in order to have a hyper expression of the molecule. So not a physiological level, but to go up above the physiological level. Since they want to correct a metabolic disease, we need as much as possible enzymes, because they are enzymes. So as this is shown on the next slide, so the enzyme is aryl sulfatase. So that disease is a terrible disease where there is progression of a neurological and cognitive disease from the age of 2 to 5 to 10, and these patients then die, and they are very severely handicapped before dying. So as you can see on the left-hand side, top left-hand side, this is the enzymatic activity measured in neutrophils. So the green line is the normal value, and you can see that in most of patients, they have more enzyme than in normal individuals. And the enzyme can also be detected in the cerebrospinal fluid. This is the right panel, which is important since the disease is mostly expressed in the nervous system. And very significantly is the clinical score of the disease. So this is the bottom panel. In gray, these circles represent the degradation of patients over time, patients who have not been treated by gene therapy. So this is the natural course of the disease. And now the colored lines on the bottom show that there is very limited progression of the disease over years. And this is even more impressive on that slide. So what is evaluated are the cognitive performances of these patients. So two forms of the disease: late onset, that means slightly less severe form of the disease, and the early onset on the right-hand side. So you can see in most, not all, but in most of the patients, the development of cognitive performance over 10 years, so it's quite significant, is normal. This is extremely impressive. And based on these results, a drug has been marketed and is available, but at a horrible price. That's another problem. Next slide, please. So now I'm moving to hemoglobin disorders. As you know, these are the most frequent form of genetic diseases, especially sickle cell disease, and also beta thalassemia. So these are mutations of the gene encoding the beta chain of the globin. Next slide, please. Just to remind you, so these are heterodimers, alpha-2, beta-2. And an important aspect of expression of these genes, and this is important for gene therapy, is that obviously we want hemoglobin to be expressed in erythrocytes, in red cells, but not in leukocytes, because we know it's toxic. So in order to do that, what has been done is to incorporate in the vector not only the cDNA encoding the normal sequence of the beta chain of hemoglobin, but also part of what is surrounded in red of the so-called locus control region. That is the region that restricts the expression of the beta globin to erythrocytes. So this is shown on the next slide. These are the vectors. I will not go into the details of what they are, but there is, of course, the sequence in green, more or less. No, not in green, I'm sorry, in purple of the beta chain of the globin, and then followed by elements of the locus control region. There are two vectors here, but doesn't matter, in order to get the appropriate expression of hemoglobin only in erythrocytes. Next slide. And this was associated with some results. It's not perfect, but this is just an example. So what is shown here on the left-hand side is the quantity of normal hemoglobin found in the red cells of a patient who had sickle cell disease. This is over time for 3 years post-gene therapy. So the good, quote, good hemoglobin is in green. So you can see it's approximately half of hemoglobin. So in this, and most of the cells in purple on the right-hand side, they have some expression of the beta chain, normal beta chain. It's okay, but it's not perfect, because in this situation, patients have less frequent and less severe complications of the disease, but still they are not cured. So this is, I will show you in a short moment a better technology, but this was something significant. Okay, so now you have to tell me when I should, where is my, I lost my, no, here he is. How much time do I have? 10 minutes, okay, that should be okay, thank you. So in vivo gene therapy with AAVs targeting post-mitotic cells, hepatocytes, the retina, the neuromuscular disease. So I will advance a bit more quickly. Okay, just one example. Hemophilia B, you know, hemophilia is, there are two hemophilias, A and B, and they lead to bleedings because the coagulation factor is on number 9 or 8 is missing. So this is an example with factor 9. So in this trial, doctors used a copy of the factor 9 gene, modified to increase its activity, actually. And this is injected in vivo, and the promoter in the AV induces the expression in the liver. So these curves show over time for 6 years even. No? So thank you. The level of factor 9 activity in the blood of these patients. And you can see that it is, no? Okay. Somewhere in between 10 to 40% of normal. It's not normal, but it's much better than 0. And so that the bleeding events of these patients is much reduced. And the good news, it looks to be stable at least for 5 years. It may be that after 5 years, it starts to decline. There are some indications, but this is an advance. It works provided that the people treated were not pre-immunized against the vector. So patients have to be selected. Only those who do not have antibodies against the vector can be treated likewise. Now we'll go several slides. Go ahead. Again. I'm sorry, but again. Stop here. Okay, I'm sorry. I just showed you one example with hemophilia B, but this has been applied with some success. Maybe the most significant success is about spinal amyotrophy, because this is the first neuromuscular disease that has been treated by gene therapy with very nice results. Now there are more than 1,000 patients treated worldwide with a stable correction, or at least partial correction, of the neuromuscular defect over years, which is good. Now I'd like to still have a few minutes to discuss genome editing, as I mentioned early. So starting with gene inactivation. Next, please. Thank you. So this is based on using the CRISPR-Cas9 system. I'm sure you know all, so I will pass that. Next slide. So here the idea is to use the CRISPR-Cas9 to induce somewhere in a very precise way in the genome a cut so double strand brain cut. The double strand brain cut will be repaired by the cell machinery of DNA repair, and this is not precise. This is the so-called non-homologous end joining repair pathway. It creates mutations, and at the end of the day, so at the bottom, the gene, if this is a gene that is targeted, will be disrupted. So it's a loss of function. So why to use loss of function? Next slide will tell you why. This has been used very nicely by our colleagues in Boston to treat, again, beta thalassemia and sickle cell disease. So the idea here is, maybe we should go one, at least maybe one back if it's possible. If not, no. Okay. So as you know, during fetal life, hemoglobin is made of alpha-2 gamma-2. So it's a fetal hemoglobin, and this is shown here. And there is a switch at birth from gamma to beta, to adult hemoglobin. So there are individuals who have persistence of fetal hemoglobin all over their life, and they do not suffer from it. I mean, fetal hemoglobin works perfectly well in adults. So based on this finding, the idea was, and this is a very smart one, is to try to block the repression of the gamma gene, hemoglobin gamma gene, in hematopoietic stem cells in order that the patients will re-express fetal hemoglobin. And this was based on the work of the group of Stu Orkin, who have identified that BCL11A is the key factor that regulates expression of the gamma chain, that represses the gamma chain. Okay, so now you can push. Thank you. So the strategy here is to use the CRISPR-Cas9 system to target the promoter that is, the enhancer actually, that is specifically working in erythrocytes for the BCL11A gene. So that if you target that, BCL11A will no longer be expressed, so that the fetal hemoglobin could be re-expressed. So it's a very good idea, and it works. We're shown on the list. This is beautiful, to my opinion. Next slide, please. So this is the first example published 3 years ago. So the patient had beta thalassemia. So in blue, this is the normal hemoglobin, the fetal normal hemoglobin. You can see that 18 months post-treatment, he has 14 grams of hemoglobin, and mostly fetal hemoglobin. And this is stable over time. Next slide, please. These are the results for sickle cell diseases, 30 patients treated. This has been recently published. For 29 out of 30, they are now free of the major complication of sickle cell disease, that is the very painful vaso-occlusive crisis. And you have, over time, up to 4 years, the total hemoglobin. And for sickle cell disease, it doesn't work as well as for thalassemia, for reasons I do not understand. But still, half of the hemoglobin is fetal, and it's stable, so it works. And next slide, please. For thalassemia, it's even better, because most of hemoglobin is fetal. This is stable. And among 35 patients treated, 32 are now transfusion independent. So it's a beautiful result. And apparently, there is no toxicity, at least up to now, of that strategy. Next slide, please. I'm almost done. So the second possible application of genome editing is gene repair, say for inherited mutations. So now, same story, to cut the DNA very precisely somewhere by the Cas9 guided by a complementary RNA sequence, but then to add a matrix here in red that can be integrated exactly at in that region of the genome and to restore a normal sequence, more or less broad. Next slide, please. So this is the idea. What has been done up to now, this is on the left side, is the classical gene addition. I described it in more detail. So now the idea is really to fix the mutation. Does it work? Well, in vitro it works. In animal model it works. But it works only in a fraction of cells. Next slide, please. So when one does that, actually one can get what is wishes, which is the 1, 2, 3, 4, the fifth line, where the pink sequence has been added, and the stretch of DNA is the normal one, and so the mutation is fixed. But in many cells, instead of getting that, you will get the number of different things that are not adequate, where the integration doesn't occur exactly at the right place, or there are modifications of the genome. So the system is not efficient enough to treat a disease. It's okay to do lab works, or you want to manipulate cells, but not for human genome editing. Next slide, please. Because of the DNA breaks, because the DNA breaks are, for some of them, repaired by this imprecise system that creates mutation. So there are alternatives that are being developed. Thank you. First, the base editing. So in that strategy, the Cas9 is modified so that it breaks only one strand, not the two strands. So there is no DNA break generated. And associated to the Cas9, to the Cas9, there is in purple an enzyme and the amynase that will modify a nucleotide, either a cytidine or an adenine, and potentially to correct a mutation. The advantage of that system is that since there is no DNA break, so there is the possibility, it reduces, that does not abolish, but it reduces the risk of ineffective or potentially harmful rearrangements of the genome. Next slide, please. Okay, can skip that. Okay. And finally, the base editing is good if you have a single nucleotide substitution. But for other kinds of mutations, more complex one, another strategy has been developed by David Liu at the MIT in Boston, where again you use a Cas9 that is active only on one strand of DNA, and you branch an enzyme that is a reverse transcriptase that will reverse a sequence which is in purple of the guided RNA to retrotranscribe it into DNA, and that will be incorporated at the right place. It works reasonably well. It looks like there is not so much abnormal sequences. So we are close to the clinical application for this strategy. Can you push? So here there are no DNA breaks, probably safer and more efficacious, but still, as I have written, the proof of concept in patients has to be provided. Now we'll go to the last slide, the conclusions. Skip that, please. Again, this stop here. So a few conclusions. So I think today we can say that gene therapy is real. It has been used effectively to treat approximately 20 genetic diseases, plus a certain number of cancer. Next slide, please. So I think we can say it's a promising, innovative medicine for many more genetic disorders, at least the monogenic ones, and maybe other cancers for immunotherapy. Next. But still, there are quite a number of issues that need to be addressed, scientific or technological. Among them, a few are to get a higher rate of transduction with lentiviral vectors. Remember what I told you about the platelets in the Wiskott-Aldrich syndrome. We need to provide automation of the production of the vectors, as well as of the ex vivo transduction. This is ongoing, but not yet fully done. For AAV, I didn't have time to discuss it in detail, but there is a lot of problem of immunogenicity and inflammation, and this is not easy to control. And the in vivo genome editing is, as I told you, not yet there fully. Next and final, there is an economical issue that is major. The prices of these products are terrible, somewhere between 2 to 3.5 million US dollars per patient. This is not acceptable, and this is a problem that will have to be addressed one day. And finally, next, these are the people involved in our studies, so the SCID and the Wiskott-Aldrich. I will just mention a few: Marina Cavazzana, Salima Asenbe-Abina. They are the two key people, and then we had a number of collaborators in many places. Thank you for your attention.
Thank you, Dr. Fischer. Nós temos tempos para perguntas. Quem quer perguntar? Helena.
So thank you very much for this very nice talk and overview on gene therapy. So I have two questions related to the CRISPR-based methods. The first is that I've seen that you skipped some delivery slides.
Can go back. Can we go back?
So my question is, what has been done till now and is working are mostly ex vivo modifications, but not yet delivery in vivo for correction. Is that right?
Not exactly. Can you go to the next, please? No, next. Okay. No. Here. Okay. Today, what mostly been done is using AAV vectors in vivo, or what is now coming but not yet used in vivo in patients is the usage of lipid nanoparticles, the same kind of particles used for vaccines. The problem with of course, it would be very elegant. The problem with the nanoparticles is that they will be trapped into the liver. So you have to modify them in order to target the tissue you want to treat. But still, there has been, if we can go back, I'm sorry, one or two slides. Here, stop. Here. This is an example, not yet published, but a first trial of in vivo base editing, where this is in vivo injection of AAV particles to target an enzyme that is involved in the cholesterol high lipoprotein uptake. And the preliminary results are rather encouraging, though some patients died from myocardial infarct, but one has to consider that these were patients extremely sick with high risk of heart disease. But so we can't yet fully conclude, but this is in vivo.
Okay, nice. So my second and very, very short question is, so the CRISPR-based strategies, they all have to be directed to mitotic cells, is that correct?
No.
Or you can get post-mitotic to work with these?
No, there is no, you can modify a gene in a non-dividing cell. There is no reason why this should be restricted to mitotic cells. I don't see any, no, it's not the case.
Okay, thank you.
Obrigado, Martim, do INCA.
Dr. Fischer, thank you very much for the presentation. Regarding the insertion of mutagenesis for the early patients on gamma retroviral vector, how long did you observe the last? What was the longest? Not incubation, but
14 years.
14 years.
14 years, yes.
Yeah, and on top of that, because there are now some concerns about also for CAR T cells on T cells. What's your, opinion or feeling about this? Because different setting, right? But it's just coming up now.
It's not the same situation as for CAR T cells, because in these cases, it was clearly shown that the integration of the vector close to an oncogene, mostly the LMO2 oncogene, was driving by transactivation, because we were using an enhancer were driving the expression, the permanent expression of the oncogene. So there is a direct link between the vector and the oncogenetic process. So what has been done to get rid of that problem is to eliminate the enhancer, and instead to add a promoter to control the expression of the transgene. So this has been done and been used since 2010. So now we have 14 years of follow-up, and so far none of the patients have developed a cancer. So it's very likely that it works. And the same system, the same technique is used for the lentiviral vectors, so the so-called self-inactivating vectors. And up to now, with two exceptions for patients with sickle cell disease who got leukemia, but leukemia unrelated to the integration of the transgene, probably for other reasons. So this is a special, there are special cases. So it looks like these vectors are safe with 10, 15 years of follow-up. For the CAR T cells, as far as I understand, this is not directly linked, again, to the integration, but to many different things, the stress to the stem cells, the immunodeficiency of the patients, the use of chemotherapy. So there are multiple reasons. And still this remains, apparently, from what I have understood, a rare event. Some 1 to 2% of the patients develop leukemias.
Yeah, that's exactly. And on top of that, because some of these events either for CAR T cells and for some of the early years gene therapy are related also for to conditioning, right? So
no, the first patients were not conditioned.
the first ones you got.
Yeah.
Okay, but now for many others,
Now, since then, virtually all of the patients were conditioned.
Exactly.
Yeah.
So there's a lot of buzz on
Sure.
this transformation,
Of
but it
course.
should be more related to conditioning.
Sure. Yeah, it's more like. Yeah.
Is there any way you can foresee that we can skip this?
Well, the dream will be, and that's also the case for allogeneic stem cell transplantation, to get rid of chemotherapy and use those strategies to displace the stem cells so that the niches are empty, and then so that the donor cells or the modified cells can enter. So perhaps one day, I'm sure you know this strategy based on targeting c-KIT or other molecules of stem cells. So to displace or to kill them without being toxic, or maybe even by using some gene therapy approaches. But this is not yet ready for use.
Okay. Thank you. Thank you very much.
Any other question?
I don't know. Thank you for your talk. I don't know if it's a basic question, but I wanted to understand why the efficacy of the treatment declines with time.
Well, it depends for which diseases. For the
You're talking about
I was
beta.
talking about hemophilia.
Yeah.
This is where there is a trend. Well, but behind that, there is an important question that is not fully solved today. That is, in hepatocytes, the AAV, or in other cells, by the way, are not supposed to be integrated into the genome. And hepatocytes are slowly dividing. So there could be a loss, some loss, although there is one site of possible integration. So it is possible that by the slow replacement of hepatocytes, the fraction of modified cells that are able to produce the coagulation factor is going down slowly.
Okay, thank you.
I have Siana.
Thank you very much. It's a very nice overview of the. I have actually not questions, but more your opinion about it. When the first issues regarding the insertional questions with the retroviral vectors came out, there was some. There were some suggestions that they could develop non-integrative retroviruses that could be the solution. I didn't see that progress much. I would like to hear your opinion about that.
Yeah, there were some vectors that were integrase defective were generated, but the problem is that there is a technological problem because the production of such vectors is very low, with very low titers. So at least up to now, this is not going to work.
Right. And the other question is, are someone looking at the possible drawback of this the therapy that is like starting to the jumping up of the heterotransposons and the endogenous retroviruses? Is there something about that? Is there
in theory, this could be considered, but up to now, it hasn't been observed in any patient. Okay. So I can't exclude that possibility. The insertions look very stable. You can trace them often in our patients. We have traced them over 25 years, for instance, the very same ones. So they don't move. Now, as exceptional events, I can't fully exclude it, but if it occurs, this will be really exceptional, so probably acceptable.
So in these patients that you developed the leukemia, not the SCID ones, but the developed the leukemia, but with no evidence of the insertional problem.
Yeah, these are sickle cell disease patients. Yeah.
It hasn't been demonstrated that was
it's not,
something
it's
related
not
to
related
that.
to transposon.
Okay.
Yeah.
Thank you.
Ultima. Ultima question.
Imagine money is not a problem. Do you think AIDA SCID patients should be transplanted every time, all the patients? Imagine money is not a problem.
Yes. Okay. let's do him. For those diseases for which we have enough follow-up in terms of safety and efficacy, I would say yes. Yes. But money is a problem. It's a very serious, even in rich countries. I mean, even in the US, even in Western Europe. And this is, I'm not sure it will be solved, actually. I'm a little bit nervous about it.
I just want to make a comment, because of all that we've heard about, biological science is essential. Because if you don't know about what the protein does, how it works, how it is expressed, we cannot work with that. And then basic science is about everything in this to get us to go further on the treatment of this disease. I think that...
Absolutely. That's a very, very good conclusion.
In clinical science, of course. Alliances, it's essential. The alliance between clinical and basic work is very, very important.
Absolutely.
Thank you very much.
Muito obrigado, vocês. I would like to thank again Serge Bob from French Embassy, who really moved forward to get this opportunity to talk with Mr. Alain Fischer.